Hello, good afternoon, good evening, good morning, everyone. I am Christian Keambou Tiambo. I am scientist at the Center for Tropical Livestock Genetics and Health and the International Livestock Research Institute, where I'm leading the Cellular Resource Functional Genomic Tools and Biotechnology program. I'm also working as ABS Nagoya Protocol Officer for the programs which are accessing genetic material at the International Livestock Research Institute. So it is a great pleasure for me to be with you for these site events to discuss on how the International Livestock Research Institute is using digital sequence information for livestock conservation and improvement. At ILRI, International Livestock Research Institute, we have one key directorate, the bioscience, which is user and producer of digital sequence information through different programs, the livestock genetic program, the feed and forage, and the bioscience Eastern and Central Africa, Baker Hill Hub, which are targeting discovering genes and delivering genetic gains. So their mandate is mostly to identify and promote and deliver appropriate genetics to the smallholder farmers in the global south, mostly in Africa, in Southeast Asia, and of course in collaboration working with other partners globally. We also have the Animal and Human Health Program, which is mandated to really manage, control, or eliminate zoonotics, foodborne diseases that matter for these smallholder farmers and the poor in the global south. And we have also in the ILRI Bioscience under the Livestock Genetic Program, the Center for Tropical Livestock Genetics and Health, which is a co-creation between ILRI and the Roslin Institute and the Scotland Royal College, which are based at the University of Edinburgh in UK, with full capacity human resource platform to develop and apply genomics and advanced reproductive technologies to address those livestock development challenges that farmers are facing in the global south. So for us to be able to produce the digital sequence information and address the challenges from farmers, mentioned by the farmers, the first thing we have to do is to ensure that everything is done in full compliance of the national and international regulations on terms of access and benefit sharing. It means the Nagoya Protocol at the local level and the international level. It means we do our work mostly in compliance or in partnership with the national system in the global south, through which we access and sample all the materials that we need to do our research, as I mentioned, from different programs here. And the material can be either directly channeled to our biorepository, where it can be kept for long-term use, or submitted to the various sequencing platform and services to generate digital sequence information through DNA, RNA isolation, proteins, and other metabolites. So all the sequences produced here are directly automatically channeled to our high-performing computer, where they are temporarily stored, and it is from there that our scientists should be able or are able to access, take time to clean, check the quality, etc., and extract the information and analyze as needed to produce what is supposed to advance farmers or the national system. And of course, when we do that, we need also to submit, because to submit them to the international platform, because as we use material available on the free access platform, IRI also contribute to those platforms by loading some of the sequences that are produced, or by loading the sequences which are produced here, to ensure that we are all on the platform to contribute to the global or the public good. Now, how do we really do it on the ground? And I'm just taking a few examples to illustrate to you how we produce digital sequence information to inform or to support national system. One of the examples is the ecological niche modeling work that was done by our colleague Olivier and Anut and partners from Ethiopia to identify the most suitable poultry genetic resources to be crop preserved in that country. It means accessing genetic material from different agroecological zones, altitude, latitude, different level of production, oxygen, different production systems, and to be able to aggregate and compute all these data and be able to inform local decision makers on which breed perform best at different locations, resisting to diseases, valuing much the locally available genetic materials, etc., or is the best preferred breed by local communities. It means it is demanded. Hence, the need for the local authorities to conserve. So this is how DSI is used also in collaboration with other sciences to improve conservation and guide improvement in the one specific location. Also, at the Center for Tropical Livestock Genetics and Health, one of our key activities is conservation of tropical poultry using the primordial germ cells. Through this system, we are harvesting primordial germ cells from blood, from blastoid eggs, from the gonad, as opposed to conservation using sperm cells. Here we can conserve the whole poultry genome, the whole animal genome in liquid nitrogens, and they are later on revived for exploration, for genetic explorations, for genetic improvement, or apply other technologies. So it is exactly what we do here, where this material which is crop preserved here can be recovered from the liquid nitrogen, and apply all the genomics and genetic tools and other knowledge that are associated to them to be able to produce what is farmer preferred or locally adapted or desired by consumers in local communities. And this is also how DSI produce from conserved material here is supporting conservation and restoration or intensive disseminations of local genetic material. And that intensive dissemination of local genetic resources can also be done through the productions of the surrogate technologies. These are animals into which we have silenced their capacity to produce their own stem cells. But once the stem cells of the biobank local chicken, as I mentioned, are injected into their gonad, they can recolonize them, and we can then use the potential of these professional layers to quickly restore and intensively disseminate the local poultry genetic resources as preferred or demanded by the national system. And here is an example of the work done at the Kenyan Agricultural and Livestock Research Organization, which is really showing interest of picking the technologies to meet the demand coming from the different countries, which at the time was experiencing a backlog of many months because Calro is not able to provide the animal seed, the chicks and the fertilized eggs that are needed or demanded. But with these technologies, the number of chicks needed could be met. The same technologies can be applied to other livestock. Here it is goat and pig, and it can also be applied to cattle, etc. Same as the induced pluripotent stem cells, which are explored here to apply all the reproductive technologies and be able to meet the demand coming from the ground. Gene editing is part of the tool which is used also then to explore DSI and be able to produce the demanded livestock. Of course, as I mentioned earlier, when this is done, it needs to be shared. It is a public good. It can be a public good. It needs to serve the community. It needs to contribute to sustainable development goals. But for that, ILRI has in place a global access policy where material collected is normally supposed to be shared to support continued research and development of tropical livestock genetics and health, and be able to serve the food and nutrition security, support the food and nutrition security in our communities. So it means what exists in our biorepository, in our high-performing computer, or what has been loaded also in the public platforms could be accessed, of course, through proper regulations to be able to contribute to the food security and poverty alleviation. Thank you very much, and it's a pleasure again to be with you, and we should be able to share and discuss on how this is effectively done, what is the experience from other parts of the world, and how can we improve mutually each other. Thank you very much.